Good morning. The purpose of this first part of the revision on molecular heredity is to fine-tune your knowledge of the first presentation. You know that we have 3 parts, 3 presentations, and this is part 1. So I found it useful to merge the content and the slides that you see with the corresponding questions and answers that might be related to the content. So we start by the first slide, and in this first slide we talk about the molecule of heredity. And of course, as you know, DNA is now known to be the molecule of heredity, and we'll be having a question about the justification of this hypothesis or this fact actually now that DNA is the most suitable molecule to represent a molecule of heredity. And this slide shows you the hierarchy. So we have DNA, and then we have genes, and then we have chromosomes. So the DNA is the molecule of heredity. The DNA makes up chromosomes, and the chromosomes are made up of genes, and we can visualize the genes and tag them with fluorescent dyes in the lab. But there is a missing item here. There is a missing item here that is chromatin and the nuclear proteins. So we will open a new screen in order to show the relationship between DNA, chromatin, genes, and chromosomes. So this question number 1, is describe the relationship between DNA, chromatin, genes, and finally chromosomes. So in interphase, as you might remember, the DNA molecule is extended like this. It is not condensed. This is in the interphase, and that's why you can see the genome as a like a chromatin thread, so, or a chromatin mass. So what is chromatin? Chromatin means that in the interphase, there is binding between the DNA molecule and the nuclear proteins, as we will see in detail after this over the 3 presentations. The nuclear proteins are of 2 types. We have the histones and the non-histones. So the DNA molecule is wrapped around the histones and the non-histones to form chromatin. So during interphase, what we see actually is chromatin. And chromatin is nothing but a mass of entangled, still non-condensed or non-folded DNA molecule that are bonded to histones and non-histones. And chromatin consists, therefore, of DNA plus nuclear proteins, almost in the ratio 50% to 50%. So when we say that in the nucleus during interphase, we see like a basophilic blue chromatin network, then we just mean that DNA has been bonded to histones and non-histones, but not yet condensed. This is in the interphase. And then in the prophase, we start seeing condensed chromosomes. So this is a chromosome. And since we can visualize it, then it is condensed, and then it has been highly folded. So what is a chromosome? A chromosome is a highly folded DNA molecule, a highly folded DNA molecule. And in order for the chromosome to be highly folded, it should be, or the folding of the DNA should be, or should have occurred with the help of histones and non-histones. So actually, a chromosome is condensed chromatin. It's a highly folded DNA molecule over histone proteins and non-histone proteins. So the chromosome is actually a highly folded chromatin. So we start by a DNA molecule. This is number one. And then a DNA molecule plus histones and non-histones result in in chromatin first, and then in very condensed chromatin, which are the chromosomes. So what are the genes? The genes are actually parts of the chromosome. So a chromosome is, if you unfold the chromosome, it is a very long DNA molecule that consists of a sequence of nucleotides, and every small or long sequence of nucleotides forms a gene. So a gene is nothing but a part of the chromosome, and therefore a part of the DNA molecule. So this is the relationship between DNA, chromatin, chromosomes, and genes. Furthermore, as it is written on the slides, and this is a very important other question, now we can identify the presence of a gene through fluorescent dyes. We have seen this in a worksheet when there was, in the revision sheet, I think, when the scientists have labeled the genes with specific fluorescent dyes. So how can fluorescent dyes help demonstrate crossing over? How can fluorescent dyes help demonstrate crossing over? Of course, usually this is not a short free response question, but a long free response question when you have genuine experiments that occurred and you have the results of those experiments and you have to deduce some facts. So for example, if I have a paternal copy of the chromosome that has a certain like homologous loci for two alleles, and we have the similar maternal chromosome, and since it is a homologous pair of chromosome, then they have the same length and they have the same shape. So this is the paternal chromosome and this is the maternal chromosome, and we locate a certain gene because now scientists are able to locate or determine the positions of the genes on the chromosomes, and we or scientists label the genes with different fluorescent dyes. So for example, here we have the red fluorescent dye, and on the other chromosome, for example, we have the green fluorescent dye. So if crossing over occurs between these two homologous chromosomes, of course, they should first form there should be a synapsis, as you know, and tetrad formation, and then there will be crossing over. So if crossing over occurs, then you will find the next step of the diagram like this, even if it's not if the sheet is not color printed, we will be writing to you that here we have the red fluorescent dye and here we have the green fluorescent dye. So again, we draw the two paternal and maternal homologous chromosomes. And of course, you can deduce that the red fluorescent dye, if crossing over occurs, then we will have here, this will stay red as it is, but the red one of the paternal chromosome goes to or exchanges with the green one on the maternal chromosome and vice versa, meaning that we will have the green maternal here and the green maternal there. So of course, here there will be written green fluorescent, green fluorescent, and here red fluorescent and red fluorescent. So this is an evidence that crossing over occurred and exchange of genetic material occurred. So this is, in order not to be confused, this is again the paternal chromosome. This is the paternal chromosome, and this is the maternal chromosome. Another important question that is related to the first slide is about the idea of the genome. So what is a genome? A genome is the total of all genes that are present in the living cell, in the organism, inside the nucleus. But if the organism is diploid, then we are talking about the diploid number of chromosomes. So in question number 3, we first need to define a genome. What is a genome? And we also need to compare, and contrast, of course, prokaryotic genomes to eukaryotic genomes. Prokaryotic to eukaryotic genomes. So first of all, what is a genome? So for example, if I have it has to be a somatic cell, because a gamete would contain half the number of chromosomes. So in a human cell, for example, in man, it should contain the total number of chromosomes, the full set of chromosomes, which is, in case of human beings, 2 times 23, which is 46 chromosomes. So a genome is the total of all the genes, the total of all the genes that are present in a somatic cell, in a somatic cell. And of course, if the organism is nucleated, then inside the nucleus or within the nuclear membrane. And we sometimes call the genome, we sometimes call it the gene pool. We sometimes call it the gene pool. But the term gene pool is rather preserved to or used in whenever we talk about a number of organisms. So we talk about the gene pool of all the organisms, like the gene pool of a community of bacteria, for example. But when we talk about a single cell, we usually use genome. Now, what are the differences between prokaryotic and eukaryotic genome? Let's remember together the differences, the essential differences between, we are still solving question number 3. So it's the continuum of question number 3. If you have a prokaryotic cell and the prototype is the bacterium, then you do not have a true nucleus, and you do not have a nuclear membrane, and you have a single circular DNA molecule that is slightly folded in a circular form, together with multiple small circular molecules that are called the plasmids, and that are replicated at the same time as the main DNA molecule. So this is the main DNA of the bacterium, and these are the plasmids. And I've told you before that some references talk about a bacterial chromosome. This is not an accurate nomenclature because it's not a true chromosome. It's not folded around histones and non-histones. If we have a eukaryotic cell, like a human cell, for example, then it should be more or less rounded, but we will have a true nucleus, meaning that we will have a nuclear membrane, and we will have inside this bright, shiny body, which is the nucleolus, which is very important for assembly of the ribosomes. And then we will have true chromosomes that are seen, at least you should be in prophase at least to see them. So what are the differences then between the prokaryotic genome and the eukaryotic genome? So we are comparing the genomes. We are not comparing the whole cell. So in case of the prokaryotes, the genome is circular. The whole genome is circular because the genome consists of only one molecule of DNA that is circular. So the genome, in case of a prokaryotic cell, is circular. The genome, in case of a eukaryotic cell, is linear. As you can see, because the chromosomes, however condensed they are more or less linear. In case of the bacterium or the prokaryote, we only have one main DNA molecule, as I just told you, one main DNA molecule that is circular. But here in the prokaryotic genome, in the eukaryotic genome, we have multiple linear DNA molecules. Of course, the number of those multiple molecules will depend upon the species. In case of the prokaryotic genome, we said that this single circular DNA molecule is not enclosed in a nuclear membrane. But in case of the eukaryotic genome, we have the chromosomes that are enclosed in a nuclear membrane. They are enclosed in a nuclear membrane, and that nuclear membrane has pores, and those pores will allow messenger RNA, that is the representative of DNA, to leave the nucleus and go to the cytosol, where it will be translated into a polypeptide. In addition to all this, we have the plasmids that are multiple. We always define them, as you know, as multiple circular. Plasmids are very important in molecular biology, so you should know how to define them properly. Multiple circular small DNA molecules that are replicated together with the main DNA, that are replicated together with the main DNA. This is the definition of the plasmids. And plasmids are considered DNA outside the main DNA. So do we have an equivalent thing in the eukaryotic chromosome, in the eukaryotic cells? Yes, because it's true that we don't have a nucleus here and a nuclear membrane, but inside eukaryotic cell, we also have DNA outside the nucleus other than the main DNA. So this is a common thing between the two, despite despite the absence of a nuclear membrane here, yet the similarity is that mitochondrial DNA and the DNA inside the chloroplasts, mitochondrial DNA, and the DNA inside the chloroplasts is considered cytoplasmic DNA or is considered extranuclear DNA, cytoplasmic DNA or extranuclear DNA. These are the major differences between prokaryotic and eukaryotic genomes, and very soon we will be seeing another difference between prokaryotes and eukaryotes, but concerning the multiplication or the replication of the DNA molecule. As you know, scientists thought at first that they didn't know at the time what the genetic material was, and they thought it was proteins because of three main reasons. So, question number 4, what are the major reasons that made the scientists think that proteins are the genetic material? Of course, this proved to be incorrect after that, but what are the arguments of the scientists to prove that, or to make them think that proteins are the molecules of heredity? Are the molecules of heredity? Actually, there were three main factors. The first one is that proteins are a major component of all cells. They are present in all cells. They are present in the cell membrane. They are present in the cell membrane. They are present in the organelle membrane. They are present in the cytoskeleton, inside the cytosol. They are present in the enzyme molecules inside the cell, and so on. So they represent a major component of all the cells. So it looks like they are something very important in the cell. They are highly variable. There is they are a diverse group of molecules. So they are important because a large percentage of the cell consists of proteins. They are diverse, they are complex, they are macromolecules, they are big molecules, which might give the indication or the impression that they must be responsible of major functions inside the cell. Furthermore, they are very specific, and therefore they do specific functions in the cell. And at the time, the last cause, the fifth cause, is that we usually are biased towards what we know. So at the time, scientists knew more about proteins than they knew about DNA, and therefore a great deal of information was known about proteins. So there are 5 reasons that made the scientists think that proteins might be the genetic material. The first one is that they constitute the major component of molecules inside the cell. They are very diverse, they are very complex, they are very large molecules, they are very specific, and we know much more about them. They knew very little about DNA at the time. And now, DNA staining, of course, as I said before several times, it might be like a question. Question number 5, why does chromatin look blue and the cytosol around looks pink? Is that important to know the color of the nucleus? Yes. When is it important? It is very important in pathology, because in pathology, the degree of staining of the molecule is important to determine by pathologists, of course, by people who are specialized, whether this is a cancer cell or not. Because if you see that there are, for example, in a cell, you see that there are prominent nucleoli and that the nucleus is deeply stained, So this means that deeply blue stain, this means, or sometimes you might have two nucleoli, and this gives you the impression that this is a very active and rapidly dividing cell. And if you see the tissue crowded with blue nuclei, because sometimes you only see the nucleus, you do not see the cytosol around, that this might be an indication of malignancy. So why does the pink color, what's the origin of the pink color? It is the stain. So we have the cytosol, most of the cytosolic compounds, like proteins, for example, absorb more or take in more the eosine stain, which is an acidophilic stain, and that appears pink under the cytosol. So substances that are acidophilic, substances that can absorb acidic stains like eosine will appear pink, whereas DNA appears blue because it absorbs the basic stain hematoxylin, which is purplish blue or bluish purple. So the chromatin, what about chromatin? Chromatin is DNA plus nuclear protein, but it absorbs hematoxylin more, and therefore it appears blue upon staining. And now we come to a very important experiment, which is Griffith experiment. What's the importance of Griffith experiment? So this would be question number 6. Of course, the title here is evidence that DNA is the genetic material, but Griffith experiment was not the evidence that DNA is the genetic material. This title applies to all the experiments that will be shown or displayed on the coming slides. So we are talking about the history of discovery of DNA is the genetic material. So Griffith experiment was the first step, but it was not the final experiment that proved that DNA is the genetic material. So what did Griffith experiment prove? So the significance of Griffith experiment, it proved only one thing. It proved that there was a substance that can be transferred from one cell to another, and that might cause a change in the characteristics of this cell. So cell number 1, cell number 2, a substance can be transferred across the cell membranes from one cell to another, and not only be transferred, but cause changes in the properties or the characteristics of the recipient cell. So this is the donor cell, and this is the recipient cell. This change that, or those changes that occurred in the recipient cell are called transformation. And the substance that has been transferred from from cell 1 to cell 2, and that caused the change, and that wasn't known at the time. They didn't know that this transforming substance is DNA. So when Griffith did his experiment, he didn't do it essentially to prove that there is a transforming substance. He did it for other purposes. He wanted to compare two strains of pneumonia, of bacteria that cause pneumonia, but accidentally, there was a very interesting observation that made scientists afterward think about transformation. And the substance that has been transferred from 1 to 2 and that caused the transformation is called the transforming material. So what is first of all, so question number 6, what was or how was Griffith experiment a first step in knowing that DNA is the genetic material? Because accidentally, one strain of bacteria, which was the recipient strain, absorbed a certain material from the donor strain and has undergone transformation or changes in property. So scientists started to study this transforming material to know what it was. But what was Griffith experiment? Griffith experiment was about two strains of bacteria, a virulent strain that causes death and a non-virulent strain that does not cause death. So in question number 7, we are going to summarize or to describe the Griffith experiment in a way that explains the process of transformation. So describe how Griffith experiment, how Griffith experiment helped scientists after Griffith, helped scientists know that there was that transformation was possible, that transformation was possible. Because this is the only result they got from Griffith's experiment. Afterward, they tried to look or try to know what the transforming material was. So we will draw a flow chart that helps simplify Griffith experiment. So in Griffith experiment, we had two strains of bacteria. Griffith was studying the pneumonia bacteria. You know that pneumonia is a disease where there is inflammation of the tissues of the lung, and it is caused by a strain of bacteria called Streptococcus pneumoniae. Streptococcus pneumoniae. And Streptococcus means simply that the bacteria are spherical bacteria that are that lie in chains. So Griffith was working on two strains of bacteria, a virulent strain that he called the R strain and a non-virulent strain that he called the S strain. I'm not going to explain the whole experiment steps because they are on the slides, but I try to summarize Griffith experiment. So the virulent strain means what? If you inject live virulent strain into a mouse, then the mouse will have severe pneumonia and the mouse will die. But if you inject live non-virulent strains, the mouse will get sick but will not die. So this is the main difference between virulent strains and non-virulent strains. So first of all, he injected live, and this is very important, which means living, virulent strains into a mouse, and the mouse died. And then another strain into another mouse, he didn't die. So this means that there was a very dangerous strain that caused severe pneumonia and a non-dangerous strain when they are alive. But now he tried something else. Well, of course, the virulent strain is considered a pathogenic strain, whereas the non-virulent strain is considered a non-pathogenic strain. And now he tried something else. He did what? He tried to kill the virulent strains. And those heat-killed are strains that were virulent strains. Heat-killed are strains that were, of course, virulent strains causing severe disease, they were injected into a medium, not into a mouse. They were injected into a culture a bacterial culture where we had the other strain, non-pathogenic, the S strain. I repeat that this time, the first time he injected the virulent strain into a mouse, and he injected the non-virulent strain into a mouse. But this time, he did a different step. He took the heat-killed R strains that were previously virulent, but of course, now they are killed. When they are killed, this means what? That they are not able to reproduce. When they are killed, they are not able to reproduce. And if they are not able to reproduce and increase in number, then if you inject those heat killed into a mouse, they will not cause death. But he injected them, or he added them to a bacterial culture that contained the non-pathogenic S strain that were still alive. So what happened is that he obtained a mixture of the two properties. The live bacteria absorbed something, a transforming material, from the heat-killed bacteria and became live but pathogenic strains. So he obtained a third strain that was totally different. He obtained a live S strain having the same cell membrane, the same cytoplasm, but that was capable of causing death. This means what? And of course, this third strain that was obtained will cause death if injected. So to summarize this, we will open a new screen because we will, this is very messy. This screen is very messy. So to summarize, we have a pathogenic heat killed, and this is very important, and I'll tell you how heat killing did not affect all the properties. Pathogenic heat killed R strain, and then they were added to a non-pathogenic. This one was pathogenic, that one is non-pathogenic. This one was heat killed, but this one was alive, non-pathogenic, live S. So what's the explanation of what happened? Why did a new strain occur? And this is the explanation you need to mention. The explanation was that there was a transforming material that was added from, or that was absorbed, transforming material or transforming substance that passed from, that was leaked from the pathogenic heat-killed bacteria and that were absorbed by the non-pathogenic live S bacteria. So heat killing resulted in what? Resulted in the fact that the R strains now have a leaky cell membrane, and this is their main DNA. Of course, the main DNA cannot pass across those leakage pores, but there are plasmids, and one of the major important characteristics of plasmids is that they carry genes that are different from the genes carried on the main DNA. And one of the most important genes that will be mentioned frequently in genetic engineering is the presence of the resistance genes, resistance against antibiotics. And another important one that is relevant here is the gene for virulence. So a very important point to retain is that genes for resistance against antibiotics and genes for virulence or causing serious disease are carried on plasmids and not on the main DNA, and therefore they can be transferred. And the other bacterium easily absorbed it without being heat treated because plasmids are easily absorbed by bacteria. So there was a transforming substance that was transferred. And this transforming substance was by plasmids that carried specific genes. Sometimes the transforming substance is not carried by plasmids, but just by DNA fragments. Any DNA fragment can pass. For example, if it happens that the main DNA here is fragmented, then it can also pass. So the transforming material and transformation can occur either via DNA fragments or via plasmids that carry genes. This is a very important piece of information to retain. And then we will be talking again about the transforming experiment because it is very important. And this is the experiment, of course. And we are going to add question number 6 because you might be wondering how DNA after heating was not damaged, how this transforming material that we now know is DNA was not damaged by heat. So here in question number 7, describe all these questions, or most of them, have appeared in a previous released official tests, or they were on AP Classroom. So describe the heat shock because we said that the virulent bacteria were heat killed. So the heat shock, we mean here by heat shock, the process of heat killing of the bacteria. So describe the heat shock in the transformation experiment. Describe the heat shock in the transformation experiment. This is the first part of the question. And the second part of the question is explain how heat did not affect the transforming material. Explain how heat did not affect the transforming material or the transforming substance. So first of all, we describe the heat shock. Why do we call it shock? Because it's like pasteurization of milk, meaning that we put in ice and then we heat. So there is this very paradoxical change in temperature. So the first step of the heat shock, how does the heat shock occur? The heat shock occurs in 2 steps. First of all, we incubate the bacteria or the bacterial culture. We incubate the bacterial culture on ice at 0 or less than 0 degree Celsius, and then we place briefly at a temperature of 40 degrees Celsius. So here is the key. We do not heat for long, and the temperature is only 42 degrees Celsius. So this is the description of the process. The justification is that this temperature, 42 degrees Celsius, and the brief duration of the heat are the key, because first of all, this rather low temperature is not enough to damage DNA, is not enough to damage DNA. Why? Because DNA is damaged at 100 degrees Celsius, at least 100 degrees Celsius. So DNA can still be transferred, can still be transferred and cause transformation. It still can act as a transforming substance. On the other hand, this temperature, as I said, causes the cell membrane to be leaky, but it's a temporary leakage. So it causes leakage in the membrane, widening of the pores. So it causes leakage in the cell membrane pores, leakage in the pores of the cell membranes, the spaces that are present in between the molecules, and this facilitates the release of the transforming substance or the release of DNA. Now the next step was to prove what the transforming, or to find out what the transforming substance was, and this is the role of Avery Avery and colleague. We call this experiment the experiment of Avery and colleague. What they did is that they analyzed they did chemical analysis of the transformation factor or the transformation substance, and they proved that it is DNA. And since DNA is the transforming material, then DNA is the substance that caused the change in the traits of the recipient molecule, and therefore it was the genetic material. So question number 8 is, describe the importance of Avery and colleagues' experiment. So the importance, these experiments, because they are very important in the history of discovery of or of proving that DNA is the genetic material, you are sometimes asked about them either in the free response questions or in the MCQ questions even. So the role of Avery and colleagues is that they did chemical analysis of the transforming material. So there was already a transforming material, and this hasn't been discovered or described by Griffith. Griffith just did his experiment on streptococcal pneumonia. So chemical analysis of the transforming material, and then they knew very little about the chemistry of DNA at the time, and they did this chemical analysis and they proved that the transforming material was DNA, and therefore, since the transforming material was responsible for changing the characteristics of the recipient cell, then DNA should be or must be the genetic material. Hershey and Chase experiment is a very important experiment in the discovery because it was further proval of the fact that DNA is the genetic material. And it depended, Hershey and Chase experiment depended on a very important fact or a very important finding that DNA, a very important chemical fact actually, that DNA contains phosphorus. At the time, they knew the chemical composition of DNA, but they didn't know the 3D structure of DNA. And this was the role of the Franklin and the scientists who came, and Watson and Crick and the scientists who came afterward, but they knew the chemistry of DNA. Otherwise, it would be impossible. It would have been impossible to do all these experiments. So they knew that DNA contained phosphorus and no sulfur. This is a very important fact. And they knew that proteins contained sulfur and no phosphorus. We are talking, of course, about the proteins themselves, not the derivatives of proteins like glycoproteins, for example, or phosphorylated proteins like the kinases, for example. So what is the scientific fact upon which Hershey and Chase experiment based their experiment? The scientific fact was this. So this is the first part of the question. The second part of the question is to justify why they used the phage, the bacteriophage virus, in their experiment. The answer is simple, because the bacteriophage is a virus that is capable of injecting its, if this is the DNA, of course, the bacteriophage is a DNA virus. It has a head, and the DNA, of course, is within a protein coat, and it has a tail, and the tail looks like a helix, and it looks like a so it can squeeze it down like a syringe in order to inject its DNA inside the bacterium, and replicates inside the bacterium, and then results in death of the bacterium, but after having reproduced or multiplied the DNA of the virus. This is not the interest of Hershey and Chase. The interest of Hershey and Chase was to know what substance is injected actually, because they knew a fact, that is, I'm now answering letter B, they knew a fact that DNA, the phage injects a substance, and that substance replicates inside, multiplies inside the bacterium. So this substance is DNA. This substance is the genetic material, and they wanted to prove this. They wanted to prove that whatever substance that is injected in the bacterium will be the genetic material. Why? Because it helps the bacteriophage multiply and reproduce. So they wanted to know what exactly was injected by the virus. Again, they knew that any substance that biologic, of course, that would be injected by the phage inside the bacterium would be the genetic material, because it helps the phage multiply. So if this substance is identified, then we can identify the genetic material. So their question was, what substance was injected? So they once labeled the phage with radioactive sulfur, which they knew would never be present unless there is a protein. And in another experimental group, they labeled the phosphorus. They used radioactive phosphorus, which they knew is only present in the DNA. How did they do this? They cultured the phage inside a medium where there was um, radioactive sulfur or radioactive phosphorus with other bacteria that were not used for the experiment, of course, in order to increase the number of phages. But every time the phage would reproduce, it would contain this material that they inserted in the culture, either sulfur 35 or phosphorus 32. So at the end of the day, they had two groups of viruses, one with sulfur 35 and one with phosphorus 32. And then they did the experiment, and at the end of the day, they found that in the first experiment, in the first group of experiment where the phage was cultured or grew in a medium with sulfur 35, they found that the injected material was cold, meaning that it was not radioactive. It couldn't be perceived by the Geiger counter, whereas the parts that remained outside which are the protein parts, were the hot parts because they were radioactive, which means that sulfur 35 containing molecules, or actually proteins, were not introduced. On the other hand, the other group showed what? The other group showed that the substance injected was hot, which means that it was radioactive, and this proved to them that DNA was the genetic material. As you know, that Rosalind Franklin was the first one to describe the 3D structure of DNA by a process called X-ray crystallography. X-ray crystallography. So the question on Hershey and Chase was question number 9, to describe the scientific principle and to describe how this led them to know that DNA was the genetic material. Of course, you know that Rosalind Franklin died before recognition of her work, and Watson and Crick are the ones who got the Nobel Prize in 1962. So she was not awarded the prize. And although they made use of the work, of her work, in their own work, yet she was not recognized for her work. So what's the scientific idea of X-ray crystallography analysis of DNA? The idea is that whenever you have a crystalline substance, this means that the atoms inside are regularly arranged. And of course, DNA, now we know that the molecule of DNA is regularly arranged. So we didn't even need to crystallize DNA because it's highly regular. But whatever other substance should be put in a crystalline form. And this means that the atoms are regularly arranged. And by regularly arranged, we mean that they are evenly spaced. And what we do is that we have an X-ray tube, a tube that has a voltage difference that generates X-rays. And you know, of course, that X-rays have very low wavelength, a very small wavelength, which indicates that they have a very high frequency. And this high frequency allows them to penetrate. See the voltage, we have high voltage here, a high potential difference between the electrodes, ranging from 10,000 to 40,000 volts. This is the potential difference that ensures generation of such electromagnetic waves, the X-rays, that have one of the very smallest wavelength in the electromagnetic spectrum and one of the highest frequencies, and they can penetrate. So here we have a LED screen, and that LED screen has a hole. Of course, a LED screen, what's the importance here of the LED screen is to prevent the passage of X-rays. Why do we put it? Don't we want the X-rays to pass to the other side? We do want the X-rays to pass to the other side, but we want to allow them to pass in a very focused, concentrated way. So here they are diverging. So we put the LED screen. The LED prevents passage of X-rays, and that's why medical doctors and technicians that take X-rays to patients stand behind a LED screen in order to protect themselves from arrival of X-rays to their bones and their bloods in order to prevent malignancy. And then here we have a small hole that allows the passage of the concentrated rays, and we want the rays to fall concentrated on the crystal. The atoms of the crystal will again disperse the X-rays, but this time in a very regular manner that represents the arrangement of the atoms in the crystal. And in such a case, when Rosalind Franklin, which was the scientist who did this for the first time, she could see here spots from the incident beams, the falling beams on the screen, coming from diffracted X-rays. So what did the crystalline solid do? The crystalline solid diffracted the X-rays, diverged the X-rays away from each other, but in a very specific pattern that reflects the arrangement of the atoms in the solid. And on a photographic plate, we obtain those white spots from the diffracted X-ray. So every crystalline solid will diffract the X-rays in a specific pattern that reflects the arrangement of the atoms inside the crystal. So this was the way Rosalind Franklin discovered several facts. So what are the facts that were discovered from X-ray crystallography? The first one is that DNA is double-stranded. The second fact was the double strands were equally spaced because from the arrangement of the dots on the photographic plate, she could see that the two strands were equally spaced. And she also discovered something genius, which is that the DNA strands were helical. So, of course, it was an amazing discovery, but unfortunately, she wasn't recognized by this. And unfortunately, none of the two scientists, or neither of the two scientists, Watson and Crick, nor the third one who was with them, who is less famous, mentioned her. They took her work and built on it. And Watson and Crick proposed the double helix nature of DNA, and they published all the data they got for this double helix nature of of the DNA molecule were derived from the work of Rosalind Franklin. But Rosalind Franklin um, had published this work earlier, especially the work on X-ray diffraction. The sixth experiment was a Chargaff experiment, and Chargaff experiment was based on biochemical analysis of DNA, analysis of the molecule of DNA, and they were the ones who explained how the double nature, the complementary double-stranded nature of DNA helped DNA be accurately repaired. So question number 11 is how can, this experiment refers to Chargaff experiment. So how can the double-stranded, how can the double-stranded complementary base pairing, how can the double-stranded complementary base pairing nature of DNA help accurate replication of DNA? How can the complementary base pairing nature of DNA help accurate replication of DNA? And I'll be opening a new screen to explain question number 11. So, of course, you know that if we have a strand of DNA and that strand of course, is a polynucleotide, as you know. So we have several diverse arrangement of bases according to the genes. Of course, some bases might be repeated. You might have more purine bases than pyrimidine bases, and so on. So if I have a strand like this of DNA, then you all know that the bases guanine, the nitrogenous bases, I would remind you rapidly of the structure of the nucleotide of DNA, because we have seen in our interrupted session that was the first session for revision of DNA, that this is a DNA strand, by the way, and we said that a DNA strand is a nucleotide, which means that it consists of several nucleotides. And the nucleotide is the monomer of DNA. It consists of a deoxyribose sugar, and this deoxyribose sugar is covalently bonded to a nitrogenous base, whatever this nitrogenous base is. And here we have the fifth carbon atom that is bonded to a phosphate group. We will be going over this description or this structure in more details in the coming over the coming slides. But this nitrogenous base, as we said last time, can be what the deoxyribose, of course, is a sugar. The deoxyribose is a sugar, and it is a pentose sugar, meaning that it consists of 5 carbon atoms, 1 prime, 2 prime, 3 prime, 4 prime, 5 prime, which is the side chain. Why do we put the prime? Because there are other carbon atoms here. We don't write the letter C for carbon atoms that are numbered 1, 2, 3. So in order to differentiate between them, we put this prime mark. So it is a deoxyribose sugar, and it is a pentose sugar made of 5 carbon atoms, and it is a deoxyribose because instead of having here OH at carbon atom number 2 prime, it only has an H. So it has a missing oxygen, and that's why it is called a deoxyribose. And a deoxyribose is the origin of the name DNA, which is deoxyribonucleic acid. The nitrogenous bases, as we said before, can be one of two groups. They may be double ringed, and in such a case, they are called purines, but they always must contain nitrogen somewhere in the molecule. You needn't know by heart the molecular structure, or they might be single ringed, and in such a case, they are called pyrimidines. The examples of purines are two. We have guanine and adenine, and guanine. So this one, the two-ringed one, can be adenine or guanine. And in pyrimidines, we might have cytosine or thymine in DNA, but we might have a third one, which is uracil, that appears only in RNA. And then we have the phosphate group. And as I said before, the phosphate group is essentially phosphoric acid. So we have PO, and then we have an OH here, an OH there, and an OH here. So it is H3PO4. H3PO4. So this rounded thing is just a symbol for the phosphate group. And the OH of phosphoric acid makes a dehydration synthesis with the OH of ribose sugar, because here this is CH2OH, and water is removed, and we have dehydration synthesis. So this bond that has been created by dehydration synthesis is called an ester bond. Because what is an ester bond? It is a covalent bond between an alcohol and an acid that is created via dehydration synthesis. You are making a bigger molecule through removal of one molecule of water. When all these nucleotides bind together, they form a polynucleotide strand, which is DNA, that consists of a ribose sugar, a phosphate group. So this is the sugar, this is the phosphate, and this is the nitrogenous base. And here we have the 5' carbon atom, and here we have the 3' carbon atom. So every time the phosphate group is bonded both sides, as I will be showing now. So here we have S and we have another different nitrogenous base, and then we have phosphate, and then we have the sugar, and then we have a nitrogenous base. And at the end of the day, we will have here a phosphate group that is not bonded. And here we have an OH group at 3' that is not bonded. But within the polynucleotide strain of DNA, you can see that the phosphate group is linked from one side to the 5' end of one ribose sugar, and from the other side it is related to the 3' end of the other ribose sugar. So what is the I'm just reviewing with you the structure of DNA because it will be it's very important to understand everything else. So inside the polynucleotide strain, as I said, the phosphate is bonded both sides. And similarly, this hydroxyl group is bonded here at the 3' carbon atom to phosphate. But at this side, you have a free phosphate end. And since the free phosphate end is always bonded to 5', then we call it the 5' end. So what is the 5' end? The 5' end is the end at which we have a free phosphate group. And what is the 3' end? The 3' end. The 3' end is the end at which we have a free hydroxyl group. Of course, what I want to highlight or to emphasize is that this part of the polynucleotide DNA strand is called the sugar phosphate backbone. And I want to remind you that all the bonds inside the sugar phosphate bone are covalent bonds, and therefore they are very difficult to break down. The nitrogenous base sequence will differ according to the genes. But coming back to our previous question, which is how does the complementary base pairing ensure accurate replication? In complementary base pairing, if I have another strand here, but I'll not draw it in detail, so you know that cytosine can form a bond only with guanine, and that guanine on either side can form a bond only with cytosine, and that adenine can form a bond only with thymine. This is what we call complementary base pairing. So base pairing means what? Base pairing means bonding between a pair of nitrogenous base. So base pairing means what? It means bonding between two nitrogenous bases, between two nitrogenous bases, bonding between two nitrogenous bases. And base pairing occurs through what? Base pairing occurs via hydrogen bonds. And this is very important for replication of DNA. And this is the coming question. How is hydrogen bonding important for replication? But let's first see how many hydrogen bonds. So this is the first time we see hydrogen bonds. So within the sugar phosphate backbone, it's all covalent bonds. Between two strands, hydrogen bonds. But there is complementary base pairing. What is complementary base pairing? معناها انه each pair of nitrogenous bases complete each other. They can just bind with each other. They cannot bind with any other bases. So cytosine can only bind with guanine via 3 hydrogen bonds, and similarly here on either side, of course. And adenine can bind with thymine via 2 hydrogen bonds. So this is complementary base pairing. So how does this ensure accurate replication? Of course, this ensures accurate replication because in replication, First of all, there is separation of the two strands. So if a DNA molecule wants to replicate, so the first thing to do is to separate the two strands. And when you separate the two strands, you expose the bases. And in such a case, every strand needs to form a replicate. So here, if those are the two strands that we have separated, then here there should have been C. And there, because to the left we have C, then to the right there should have been G, and here T, and here A, and here T. So those are the two strands that we have separated. How have we separated them? By breaking the hydrogen bonds. And this is the important question. I don't remember, it might be question number 13. What is the importance of having hydrogen bonds for replication? Because the hydrogen bonds are weak bonds. And if they are weak bonds, then they are easy to break. And breaking them is important because we need to separate the two strands. And why do we need to separate the two strands? To expose the nitrogenous bases. And why do we want to expose the nitrogenous bases? We want to expose the nitrogenous bases so that there could be easy accessibility to the enzymes of replication. So every time the enzyme of replication, say this is the polymerase enzyme of replication, every time it finds G, it will get a C. And every time it finds C, it will get a G. And every time it finds A, it will get a T. And every time it finds T, it gets an A, and here T. And then it will link them to the sugar phosphate backbone and form, via the ligase enzyme, form a new strand. So this is how complementary base pairing, that has been proved. All this is related to Chargaff experiment because complementary base pairing was the discovery of Chargaff experiment when they analyzed the chemical structure of DNA and found out that for stability of the DNA molecule, G should base pair to C via 3 hydrogen bonds and A should base pair via to T via 2 hydrogen bonds. And we also tackled another question, which is what's the importance of the presence of hydrogen bonds between the two strands of DNA. We will stop here and then I'll be sending another part of revision 1 of DNA.